Hello and welcome to a brief introduction to the research in my laboratory. As most of you know, our tissues are composed of very diverse cell types. And similarly, when we conduct experiments in the laboratory, for example, when we differentiate stem cells into different cell types, we generate fairly heterogeneous mixtures of different cell types. This is quite important, for example, in the context of directed stem cell differentiation. If we wanted to take this gray stem cell and convert it into a desired cell type, in this case here shown with blue, we typically find that doing this in the laboratory results in very many cells that fail to differentiate into another cell type. It results in cells from different cell types and so on. And optimizing such directed differentiation protocols is usually very challenging and based on trial and error approaches. If we could make single cell measurements to identify what are the signaling mechanisms resulting in those different branches, then we could use a more rational approach to either stimulate or inhibit different signaling pathways so that we can more efficiently generate the targeted cell type of interest as shown here. And furthermore, we can use those single cell measurements to estimate how accurate and how precise the differentiation protocol is. This type of experimental paradigms has motivated a great interest in the development of single-cell methods, and recently single-cell RNA sequencing has made tremendous progress in that direction. As useful as these RNA measurements are, however, they're not sufficient to characterize what's going on in different cells. And as a particular example here, I point to the well-studied protein p53, the famous cancer tumor suppressor gene. And if we measure its abundance at the transcript level or the protein level across a cohort of cancer patients, we find that the correlation is rather poor. And if you see poor correlation between RNA and protein, you should always suspect that this is due to measurement error. In this case, this is not what's happening because decades of extensive studies of p53 have demonstrated that the expression of p53 messenger RNA is constitutive, while the abundance of the p53 protein is mostly regulated by protein degradation. Therefore, we don't expect to have high correlation to begin with. And similarly, when we took a much more comprehensive, unbiased, and systematic exploration for the abundance of proteins and RNAs across different human tissues, here on the right, as shown with this cartoon schematic, we found that generally RNA abundances can predict whether a gene is highly abundant or lowly abundant across all tissues, but RNA levels tend to be less powerful in predicting how a particular gene, for example, gene 5, will be changing across different sets of human tissues. And this motivates the need to perform not just RNA measurements, but also protein measurements. And usually such protein measurements are performed using antibody approaches, but they afford only limited specificity, and they have allowed quantifying relatively few proteins per sample. So we were interested in developing mass spectrometry-based methods that can increase both the specificity and the throughput of quantification so that we might be able to specifically quantify many thousands of proteins across many thousands of single cells. Towards, and just as an illustration of the low specificity of antibodies, I'll offer to you this statistic published recently in Nature, indicating that annually we spend over $1 billion on purchasing antibodies with poor specificity. And this figure does not include money spent on following flawed hypotheses This is simply money spent on purchasing antibodies that do not have the specificity indicated on the label. To realize single-cell protein analysis, we have, by mass spectrometry, we have developed both experimental and computational approaches. Experimentally, we have developed new types of sample preparation that do not require detergents and that allow to both miniaturize the volume of sample preparation and fully automate it so that we can process thousands of single cells with limited batch effects and limited amount of effort. We have developed the isobaric carrier concept that allows us to more efficiently identify peptide sequences and reduce the losses. I will not explain the isobaric carrier concept here due to lack of time, but you can go to our YouTube channel as indicated here in red at the bottom and find many videos where this concept is explained by us and others as part of the conference that I organize or presentations that I have given for our work. I'll also mention that this type of approach requires substantial developments on the side of data analysis and interpretation, and my group has developed such tools. For example, a graduate student, Gray Huffman, developed DoMS, which stands for Data-Driven Optimization of Mass Spectrometry Methods. It's a set of tools that allows us to visualize data and determine optimal parameters for our analysis. Similarly, we have developed methods such as DART-AD that use Bayesian frameworks to incorporate additional information for peptide sequence, such as retention time, to again enable us to assign sequences to larger number of peptides. We have applied our single-cell proteomic methods to a variety of different systems. One particular example that I'll mention here is understanding macrophage polarization. Macrophages are innate immune cells that are present in all of your tissues. They're part of the initial response that your immune system has to various pathogens. They're part of regenerative processes, and they can have very different phenotypes. They can either be pro-inflammatory, stimulate inflammation, or they can be anti-inflammatory. And those different phenotypes has major biomedical ramifications. For example, anti-inflammatory macrophages are sometimes known as tumor-associated macrophages that might prevent your immune system from effectively eliminating tumor cells. In my lab, we are also interested in mechanisms of post-transcriptional regulation, and specifically, we are very interested to understand how modifications of ribosomes can contribute to regulating protein synthesis. This is a new and largely unexplored mechanism for regulating translation of messenger RNAs. And we have focused on studying this mechanism also in the context of immune cells, and specifically we have very exciting data looking at how ribosomes remodel and affect protein synthesis in the context of monocytes differentiating to macrophages. And in those model systems, we can isolate ribosomes directly, physically, by using sucrose gradient sedimentation, as shown here on the bottom. This is a classical technique that allows to sediment different parts of the cell based on their size, based on how quickly they sediment in sucrose gradient, and then to analyze them as we have done downstream using mass spectrometry and other methods. Our work is very collaborative. Here you can see one picture of the team that we took recently before the single-cell proteomics conference that we organized in the summer. And we have been fortunate to be supported by many funders, such as the NIH Directors Award, the Paul Allen Frontiers Group, as well as local biopharmaceutical companies such as Sanofi, Merck. We are also supported by the Chan Zuckerberg Initiative, and we are part of the Human Protein Atlas. I hope this gives you an overview of some of the research projects going on in our group that are carried out by our team. And if you're interested in learning more, you can visit our YouTube channel or our website and delve in, learn about the research that keeps us excited.